This is a production of Cornell University.
So yes, so that's the title of the presentation. So I will, let's just put my timer here. So I will, that's the outline of the presentation. And again, if you have any issues hearing me by any chance, maybe internet connection, just please let me know, Jennifer. So go a little bit of terminology and a little bit talking about enteric methane emissions and measurements. Then show some data from meta-analysis that we did here last year that compare grazing and confinement. Then some dietary strategies to mitigate methane emissions in grazing systems, but also some of them actually more at this point apply to confinement system, but also potentially to be used in dairy, in grazing, and some conclusions, future research, and questions. In terms of terminology, just I know that some of you may not be familiar, but when I'm referring here to confinement, confinement can be year-round, meaning cows can be on barn the year-round. Like for instance, most of the dairies in the US, they are in a confinement system. Confinement can be seasonal. Let's say pasture-based dairies here in the Northeast and the Midwest, they are seasonal, meaning cows are confined at this time of the year because there is no grass to graze. And then during the summer, the grazing season, they are out grazing and they receive different types of supplements, right? Or full grass diets. I'll be talking about total mixed ration, or TMR. Basically is a ration where you mix your forage source like hay, silage, or haylage, your grain source, and then minerals and vitamins. You can use a vertical mixer like you see here in this picture and mix all together and offer to the animal, right? So a TMR, most of the confinement systems in US, dairy system feed TMR, but you can also have a grazing farm where the cows go out and graze, and then when they come back for milking and after milk, they can get a TMR. So TMR is not a synonymous of a confinement. You can feed TMR to a grazing cow. So I'll be talking about dry matter intake. That's equivalent to feed intake, right? It's dry matter, for instance, because if you want to compare a pasture source, which is wet, right? Like 85% dry matter, and a hay, 85% moisture in the hay, that's the opposite. So to compare this on a fair base, so you use a dry matter system, meaning you dry the samples in 100 degrees Fahrenheit or 55 degrees Celsius for 48 hours. You remove most of the water, and then you can compare the nutrient composition on a dry matter base. And then we're talking about energy corrected milk. Basically, you're normalizing here the energy of milk on a assuming 3.5% fat and 3.2% protein. In this way, you can compare a Jersey cow, right? Those brown cows that produce about 5% butter fat with a Holstein cow that produce about 3.5% fat. You can equalize, you can normalize milk production on an energy base. A little bit of other terminology here. When I'm referring to methane production, it's basically the absolute amount of methane that's produced by a ruminant animal, and then methane yield. And then we see a lot of methane yield, and intensity basically is grams of methane either per kilogram of dry matter intake or DMI, or per kilogram of energy corrected milk. Right? Again, you're kind of trying to normalize things here to make those comparisons. Just to give a very quick example here, where you have a scenario of a cow A, let's say, assume that she produced about 500 grams of almost like a half pound, more than a pound actually, of methane. This cow is consuming about 30 kilos of dry matter, and she's producing 45 kilos of energy corrected milk, about almost like 100 pounds of energy corrected milk. So you can make these calculations and come up with the numbers here. Methane yield would be about 17 grams per kilogram of dry matter intake, and methane intensity about 11.1. Let's say this cow B, she produced the same amount of methane, but she's eating less, and producing less milk. So you can see that the methane yield will be high. Methane yield will be about 20% higher, and methane intensity would be about 29% higher, right? So you can have an idea here how, when bringing those numbers to a normalized base is different. So I guess what I got here
For a
on
second, sorry to interrupt you. You are sort of going in and out the sound.
Yep.
I don't know what's causing that.
Is better now?
Yeah, you sound good now.
Okay, maybe I'm moving too much. Okay, I'll try to be close to the computer. So I guess the question is why a shark is doing, and again, Jennifer, if you see this problem again, then I can try to put my another sound system here. So so what a shark is doing here, right? So, and this is a tiger shark. Well, and probably not eating a cow. They probably not cross path by any means. But the thing is, a cow or ruminants may be affecting their environment, right? So in this, there is effects of climate change on shark migration. This is a very recent study that was published actually this year at the Global Change Biology, where the researchers are estimating based on modeling and also tracking these animals that for every degree Celsius of warming temperature in the oceans, they have been tracking this for about 10 years. Those animals are basically extending their migration further north, right? 250 miles further north. And also they start their whole trip summer migration 14 days earlier because of the warmer temperature. So you know what this would have impact in the whole ecology of apex predator in the web chain, food chain, you know, is unknown, right? So that's why we're focused on methane. So and just complement what Larry already mentioned. So a cow as a ruminant, and this is not only a cow, ruminants in general that include sheep, goats, wild ruminants like deer, moose. Those animals, they have one stomach with 4 compartments. And the 4 compartments are shown here: the rumen, the reticulum, the omason, and the abomason. The rumen, reticulum, and omason, it's an aerobic environment with billions of microbes, like Larry mentioned. The abomason is equivalent to our stomach. Basically has the same functions of a stomach of humans. It produces enzymes that mostly digest protein, right? So, and here's just a view of the full rumen. You can see it really, the space that Larry was mentioning is very huge fermentation vat, all the left side of the animal. You can see the reticulum right here, right? And just for a reference, the heart is right here. And as I said, there are billions of microbes from protozoa, methanogens, the archaea, and also fungi and yeast. A little bit of the rumen fermentation, again, to complement what Larry said. So these animals are consuming feed, and this coming from a forage source like silage or hay, or grain source like corn, for instance. So you need to think this basically can divide it in 2 major carbohydrate source: fiber and starch. The microbes will attack fiber and then starch and start to degrade, right? And start to decompose and release glucose. Another set of microbes coming in and use glucose as an energy source, right? And they produce what Larry mentioned as a microbial protein. So microbial protein will be a major source of protein when it passes through the small intestine, right? So, and get digested and release amino acids for those animals. It's a high quality protein and is manufactured by the microbes because of the symbiotic relationship with the ruminant, right? So the glucose, and then it's further fermented, and there is byproducts of glucose fermentation, right? So hydrogen, like gases, CO2 and methane, and also the VFAs, of volatile fatty acids, which are acetate and propionate and butyrate. So those would be absorbed to the rumen wall, right? And then goes to the blood, and in the liver will be utilized. One thing that's important here, acetate is used for fat production in the mammary gland for making butterfat. Butyrate is also for fat production, and propionate is used for glucose production. So in other words, the ruminants basically use those byproducts of microbial fermentation to make milk, like Larry mentioned this extensively. So nothing that Larry mentioned that's also showing here is you have this positive correlation between dry matter intake and methane production. In other words, as intake goes up, enteric methane production also goes up, right? This positive correlation, this equation here, coefficient of correlation of 0.86, pretty high, and that's why it comes down why we should mitigate methane. The reasons, one, the obvious is reduce the carbon footprint of dairies, but also methane represent energy loss, like Larry mentioned as well. So this could be between 3 and 10%, like in this study done by New, but it can range from 2 to 12%, depending on where you looked at. But the point is methane also represent energy loss, and that energy that's not captured into milk production, right? Because it's lost through burps. And that's very important to identify this. And this is a kind of a joke here, but the point here, 95% of the methane is burped, right? And only 5% comes to the other end via flatulence. So the point here to say some of the technology that is being used now to measure methane focus only on the 95%, right? That's I'll show in a minute here. So to finalize this process about methanogenesis and enteric methanogenesis, so like Larry mentioned, methane it's basically synthesized by methanogens, or the archaea, the microbes that are in the rumen. You can see them here, these blue cells here that are attached to a protozoa cell. They have also symbiotic relationship. But methane is used as a CO2 and hydrogen. The microbes use CO2 and hydrogen to make methane as substrate, right? In the metabolic pathway of acetate production, one of those VFAs, right? And you can see that hydrogens are released when you produce acetate. So in other words, diets that are high in cellulose or in fiber, usually you have more methane because the pathway leads to more production of acetate, and acetate leads to more production of hydrogens, which is a substrate for the methanogens to produce methane. On the other hand, in the metabolic pathway that produce propionate in the rumen by the microbes use hydrogen. So it's a hydrogen sink, like Larry mentioned. So like starch-based diets like corn, usually you have more propionate production or increased propionate production, which use hydrogen. So that means less hydrogen for the microbes to make methane. That's why a more grain-based diet or supplemented diet that has more grain, usually you reduce methane, has less methane production than diets that are very high in forage. And here are some of the ways that we can actually measure methane. You can see here, that's what Larry has mentioned, like a respiratory chamber that's being installed in Cornell. So that's considered the gold standard for methane measurements, because you can see it basically, the cow is in the chamber, all the gases, in not only the rumen to the burps, but also to the flatulence, the flatus, all this gas are actually measured, right? So the downside of this equipment is the fact that when the cows are inside, of course, that can affect their intake. And if it affects intake, it can, bias methane production. But again, I still consider the gold standard. They're now using this kind of either panels here that the cows can see each other, because in the past they didn't used to have those, so that's important, so the cows can at least see each other. And another small issue of this system here, you keep the animals there for a couple of days only, 2 or 3 days, just because it can affect the intake, and you don't want that happen. So there's some new technologies like the green feed system that is shown here that we use here at the University of New Hampshire. Basically, the system functions like you have a cow. It's basically a head chamber, right? You basically, in this case, you only capture, not only, but capture most of the methane, about 95% of the methane that's produced by the animal, capturing and measuring, right? It's coming from the rumen and via burps. The cows wear RFID in the ear, which is basically a tag that triggers the system, which then automatically release some grains. So while the cows are eating grain and burping and eructating, basically releasing gas, so there are sensors here that can actually measure methane and CO2 and even hydrogen and oxygen. So there is a head position sensor that maybe the cow is too far away, will not take the measurement, so the measurement is very reliable, right? So the good thing about this system is you can have animals visiting like we have here in our dairy, in our organic dairy. So cows can come and visit like 5-6 times a day, right? So you have several measurements during the day, and you can do during multiple days, right? So you can, like we usually take measurements in a study from beginning to the end that may last up to 80 days, 90 days of measurements, right? So we have more numbers, right? More spot measurements. Here's the setup of the same equipment for grazing. So you can also have the system set up for a grazing. You can see the solar panels. So what you do here, we have the cows on pasture visiting, and when we move cows, we, in our system, have a rotational grazing. As the cows move, we keep moving the equipment together with them. So it's a very nice system. Just another version of the system that Paul Kononoff at University of Nebraska used quite a lot is called the head box system. As you can see here, kind of similar type of technology, I would say. And in this case, it's basically cows needs to be in a stall, but you can see, you basically capture methane that's produced in the rumen, right? And then you also have this. It used to be very common, now it's less, with the green feed coming into the picture. This is the SF6, or the extra fluoride tracer technique, or this we did here at UNH as well. Basically have this tubing here that goes close to the nostril of the cow that capture the gas, and then we basically, and the gas is accumulated here in those canisters. You remove the tubing here, get a syringe, get a gas sample, and then you put in you analyze in the gas chromatograph for methane, right? So when you use the tracer, because at the same time we have SF6, which is basically a capsule that the cows will swallow and release SF6. So we do a ratio between SF6 and methane, and come up with the calculations. So that's another way. You can also use the system like this for cows that are grazing, right? So, I talk a lot about methane, and just going to quickly mention here, if you want to see the whole farm emissions, you need to consider also nitrogen. I'm not spending too much time talking about nitrogen here, but when you're talking about nitrogen, like Larry said, a lot of those diets nowadays in the US, we have been reducing a lot the nitrogen concentration, the protein of those diets. However, pasture-based diets, just by default, usually are higher in nitrogen because of grass, specifically if the grass is mature, immature, early in the season. Lush pasture has very high levels of nitrogen. So in those situations, you end up having an unbalanced supply of energy and nitrogen in the rumen. And when this happens, so you have more of the nitrogen basically leaking out. In other words, you see a substantial amount of nitrogen being excreted in the urine in the form of urea that's very reactive, right? Some nitrogen going out in the form of feces or fecal nitrogen. And you have a lower proportion of nitrogen being converted into milk protein. That's what we want, right? We want to see as much as possible dietary nitrogen or dietary protein being actually converted into milk protein. That's the goal. So when you have too much nitrogen in the diet, that's more difficult. And a lot of this nitrogen got lost, right? And convert into nitrates that can contaminate water and soil. You can also have other changes, like some of the nitrogen can be converted into ammonia that is pose some risk for human health, and also, I'm sorry, and also for nitrous oxide, right, which is a major greenhouse gas, right? So even though I'm not talking much about the nitrogen side here, it's important to look into that as well. So I cover a little bit of the terminology, enteric methane. Now I'll get into a little bit of the comparisons between grazing and confinement systems. What we did here, before I get into this, the question is why grazing, right? So, well, grazing prefer, multiple ecosystem service, right? From pollination, erosion control, nutrient cycling. Now, the unique aspects of grazing is the milk, right? Milk from cows that are on grass usually have higher concentrations of omega-3 fatty acids, which has been linked to better human health, right? That's it's well proved, and they have more omega-3 fatty acids in cows that are consuming TMR, right? So also pasture-based systems are perceived to be more environmentally friendly, more animal welfare friendly by consumers. Pasture also is a very cheap, is probably the cheapest feed source. So if you have more pasture in the diet, that's can probably benefit the bottom line there in terms of economics. So overall, there are outstanding opportunities for market capitalization on pasture-based dairies, specifically, grass-fed dairies right now, right? Grass-fed dairy, grass-fed beef, right? So the products are more, you pay more, the farmers receive more for those products. There's some issues, not say issues, but necessarily things that you need to consider on a dairy, on a grazing farm. One, we know if you look at here, this comparison is study from Fernando Bargo from Penn State. They looked at the diet that was TMR, cows were on confinement getting TMR. They're grazing, but supplemented with TMR, total mixed ration, or they're grazing and getting some grain, some concentrate, right? And you can see that the intake that cows that are grazing are usually lower than intake that the cows are on confinement, receiving TMR. Therefore, milk production also goes down, right? You see that grazing, cows that are grazing and supplemented TMR has less milk production, and cows that were grazing and receiving grain, they're also producing less milk. Of course, this can have implications on economics of a given dairy, right? Another aspect, and this is maybe it's kind of, I think it's obvious, is the cows that are grazing, they spend more energy because they have to walk and they have to basically feed themselves, right? So that's why in this study here, they compare the confinement, and that's the energy expenditure in kilojoules per kilo of metabolic body weight. And you can see the cows in confinement, you spend less energy than cows that are grazing. That's, again, obvious reason here, just because cows that are grazing, they need to move, right? And basically get feed. Of course, if they spend more time grazing, more energy to graze, so less energy is available for producing milk, right? That's some, and of course, they spend 21% more energy basically to graze. So, and that energy is not going to milk production. That's why milk production usually decline when cows are grazing and also feed intake. So why confinement? Well, usually have more dry matter intake, have more milk and milk components, usually have better feed efficiency, cows has better body condition, cows are not exposed to inclement weather. Sometimes it's even in the summer, you know, weather can be really difficult for the cows, specifically heat stress in some parts of the country, even here now in the Northeast. So confinement, you know, you have those advantages, right? So we came up with idea here with my grad students to kind of build up a data set to compare feed intake, milk production, and methane emissions in dairy cows that were on confinement or grazing, right? So it's kind of difficult because there's not a lot of studies that has done that, meaning making those comparisons. And what we did here, we have this flow here to select the papers that has been published in the literature. We identify them, we screen them, then we choose papers that are eligible and then include or not in the data set. So we start with a total hits of 1,000 papers and we end up with only 8 studies. And why we have only 8 studies based on the literature is because, again, what we're looking for were studies that simultaneously evaluate in the same study, right, a treatment or a diet that cows were in confinement and a diet or 2 diets that the cows were actually grazing. And not many studies have done that. So, and also we eliminate papers where, for instance, if they don't report dry matter intake or individual dry matter intake from cows on pasture, we did not include those papers. So we end up having short numbers of papers. We end up with a total of 10 because there are 2 studies that we did here at UNH that are not yet published, but we included in the data set. So we end up with a total of 10 studies. So, and the way that we should be looking at this forest plot here, it compares the mean difference between the confinement and the graze. What you should be looking here on the left side is all the studies that we include in the data set, and you see all the comparisons, but the focus here should be in this diamond here in the bottom. That shows that on average, right, in terms of intake or dry matter intake, cows that are grazing, they consume about 2.5 kilos less feed, or about 5 pounds less feed. And this was, you know, statistically significant, right? So again, going back to what we discussed, so cows that are grazing, they need to walk to find their feed, right, the food. When you look at milk production, the same trend here, right? You see the diamond here in the bottom, that's the overall average difference. And cows that are grazing, they basically producing about 3.6 kilos less milk, or about 7.5 pounds less milk, right? And the same goes for milk fat production. They're producing less milk fat, and they are also producing less milk protein, right? So of course, this can have implications on, as I said before, on the economics and profitability of those dairies that are on the grazing. Even though pasture is the cheap feed source, you know, the economics may play a role here when you drop milk components, because farmers are paid based on milk components, right? So that's something that needs to be evaluated and as we move along. So we also estimate and methane production between confinement and grazing. One thing that I want to highlight here, folks, be a little bit careful when looking into this data, because only few studies actually report methane production. Most of the studies did not measure methane production. So what we did, we estimate methane production using an equation from New et al. In 2018. So you need to be careful when looking at this data, but there's, as I said, a very limited information out there. So what we did, we use the equation and we estimate methane production, and you see that the cows that are grazing, they produce less methane on a absolute amount, absolute base. This is it's be expected because they consume less feed and then they produce less methane. We did not see a difference, right, on methane yield. That's grams of methane per kilogram of dry intake. It's the p-value of 0.83 here, but we saw a trend in methane intensity, which is grams of methane per kilogram of NH-corrected milk. So cows that were grazing, they tend to be less, to produce more methane per kilo of NH-corrected milk. And a p-value of 0.008 is a statistical trend. It's not significant, but it's a statistical trend. When you look at milk nitrogen efficiency, that means the higher the number, the better, because that would indicate that more of the dietary protein is converted into milk protein, right? And that's what we want to see. And even though the grazing was less efficient than the confinement, this was not statistically significant. Again, if you have more studies, more power, maybe those numbers will be different. But with the number of studies that we use, only 14 treatment mean comparison, that's a low number. So, you know, folks need to be careful when interpreting these results. But it's a start here on this process. So I basically discuss a lot about individual cow production data, and now I'm trying to move more into whole farm emissions, trying to do some comparison here using some the integrate farm system model from those two studies. There is studies from Victor Cabrera lab and also for Erin Silva, both from Wisconsin. What they're looking here again is the integrated farm systems model that Larry also shows some data from Cornell, from New York farms. And again, this model takes into account several aspects of a farm, right? From the harvesting, from the milk production, from fertilization, carbon sequestration, and so forth. So it tries to, is almost like a life cycle analysis from our rots that developed this model. And I was showing here some data from Wisconsin dairy farms. Those were organic dairy farms based on a survey with 69 organic dairy farms. So based on this survey, they were able to make those clusters, which basically farms that are either they look like or they're different, and they group those farms in 4 groups. And you can see here some of them have higher milk production per cow per year, like clusters 1 and 2, and some have more feeds, use more feeds than others. But what you can see here, the major difference between those clusters was basically cluster 1 and 2, where cluster 1, they have more feeds and feed more grain, while cluster 2, so they basically have, cluster 2 will have more pasture in the diet, right? So this kind of white portion here is equivalent to pasture. They also have a little bit of concentrate, but more simple diets compared to the cluster 1, where they have hay, corn, silage, also concentrate, and other feed stuff. So those are the major difference. Cluster 3 and 4 were intermediate between those two. So what they did also here was to run the model and compare those dairies in terms of the whole farm greenhouse gas emissions in kilograms of CO2 equivalent per ton of NH-corrected milk. And what they found out was that the cluster 2, which is the one that has the highest proportion of grazing, or the more time of cows on pasture. As you can see here, that's the grazing time on pasture in a year base, right? Ranging from about 140 days up to 190 days. Those cluster 2 that have cows for longer periods on pasture was the one that produced more greenhouse gas emissions, right? right? So based on that model. It's another study that also did some comparison like this, looking at grazing intensity of farms also from Wisconsin. And they have different grazing scenarios from G1 to G5, G1 being the ones with less grass grazing in the diet, about 9% of the total intake, and G5 be one of the highest amount of grazing in the diet. But overall, they're somewhat comparable. You can see the methane, again, it was in blue here, basically it's the highest source of greenhouse gas. It's not a surprise here. That's based on what we have been discussing. But what I want to highlight here very quickly is when they add grain in the diet, when there's more grain in the diet, like in the G2 scenario here, so they have more grain, they drop methane. The same thing here with these other scenarios. When they add more grain, like G5 here, there's more grain in the diet, so it dropped the whole greenhouse gas emissions. And then the two confinement systems, the C1 and C2, those confinement systems either have an aerobic digester or not. When they have the anaerobic digester, you can see that it dropped also the greenhouse gas emissions, mostly because you reduce manure methane, right? That is emitted via manure. So that's, again, that's a possibility, that's a good thing, and is in line what we have been discussing here. Right. So we discussed about terminology, did some comparisons. Now I'd like to move on and talk a little bit about some dietary strategies to mitigate methane. So there's several ways to do that through forage quality. I think Larry mentioned a little bit of that, and I will be exploring a little bit. Forages, secondary compounds, things like condensed tannins, glucosinolates, condensed tannins, some legumes like bird's foot trefoil had higher levels of condensed tannins. I'll talk a little bit about glucosinolates. Supplementation strategies like feeding grain, feeding oil seeds, not mention this today because of time. Some feed additives like monensin, nitrate, or nitroxypropanol, 3-NOP. I'll mention a little bit of that today, and also seaweeds, which is another option as well. So, and starting with the forage quality, we have been doing some studies with brassicas, right? So brassicas include things like kale or canola, and they are very winter hardness. They can stand very low temperatures. For that is good because you could potentially extend the grazing season, and they can also be used as a cover crop. There are data out there showing that it can reduce methane emissions. These are data that was done in vitro in the lab. High biomass and high nutritional value. So this is a good candidate, right, that we have been looking at. So we did a study here at UNH where you have cows, we used 20 organic cows, either the cows were in a controlled diet, so 10 of them in confinement all day long, and the other group where cows are half of the day in the barn receiving a total mixed ration, and then they move to graze the canola, right? So that's how we set up the study, and this was part of that meta-analysis that I mentioned. Just to give an idea very quickly here, the diet has about 6% forage, right? The control diet, the canola diet, has the same level of forage, but to replace some of the baleage, some of the hay with canola in the diet, right? As a grazing source. And we allow, the calculations were those cows would receive about 42 pounds of dry matter from pasture from canola. This is just the layout of the study. You can see the barn is here, the cows walk to the grazing areas, and every strip here represents a strip of forage canola daily that was offered after the milking, after the afternoon milking, right? And you use two fields, these two adjacent fields here to complete the study. Again, we use the green feed system. I mentioned this to actually measure methane. When you look at methane production, and we did the measurements on week 3 and week 5 of the study, you can see the methane production decreased by 35% when the cows were grazing the forage canola, which was, important reduction. Methane yield decreased by 29%. You can see here, cows that are grazing canola has lower methane yield, which is grams of methane per kilogram of dry matter intake, and methane intensity decreased by 28%, again, grams of methane per kilogram of NH-corrected milk. So those are kind of exciting data here based on this forage canola. So why made that happen? I said, as Larry mentioned, so high quality forage, and that's happened with canola. So you can see here the crude protein and NDF, which is basically the fiber, crude protein is high, ranging between 20-27%, and fiber is low, right? So it's highly digestible. Lots of sugars, which is fermentable energy, right? So that may explain why methane was reduced, right? Another aspect is the rumen VFAs that we have been discussing and I mentioned earlier. So acetate, right, drop on the canola diet, as you can see here, and propionate went up. Remember that propionate is a hydrogen sink, removes hydrogen from the system, and acetate gives out hydrogens, right? So less hydrogen in the system means less methane. And another thing that is going on here with canola, and this, I want to mention this very quickly here. This was a study that was done in Kathy Soder lab. So they have this simulated rumen system, meaning this is a simulated rumen, as you can see here in those bottles, right? And they take rumen fluid from a cannulated cow and then do some inoculations with buffers and so forth and create an environment that's similar to the rumen. And they test that in that system, and they saw a negative correlation between methane emissions and glucosinolates. Glucosinolates is a compound that's relatively high in brassicas, and there is different types of glucosinolates, as you can see here, this kind of fancy names here that I'm not trying to mention here to pronunciate, but you can see that that there is negative correlation with some of them, like this glucobrassicine here, meaning more of this compound, less methane, and they also see this other one here, glucoraphanin, and the total. So overall, there was a negative correlation, more glucosinolates, less methane. So this may explain why methane dropped when my cows here at UNA graze canola, right? Still on this high quality forage aspects, this is another study that was done in Ireland. And what's interesting here is that, so either cows was fed in TMR in a confinement, or they graze in perennial high grass, which is very high quality grass. So they saw a drop in methane of 27%. In methane production, methane yield dropped 12%, and methane intensity dropped also 12%. So again, why? Well, perennial or high grass, ryegrass is very high quality grass. And if it's on early maturity, like you can see in a rotational grazing system, so you can see the crude protein is high, 24%, and fiber is relatively low, right? 46% NDF, 24% ADF. Very high digestibility, 83%. So when you combine this, that you may see why methane production will drop when you're comparing cows grazing perennial high grass with a TMR diet in confinement, right? So there's some options there. And now I want to move to seaweeds. I have another 5 minutes or so here. And this is a couple of studies that has been done in Penn State and another one in UC Davis. So the study that was done in Penn State with this red seaweed called Asparagopsis taxiformis, and they fed different amounts from 0 to 0.5% in the diet, not a lot, right? And they saw substantial reductions in methane production. If you compare, the 0% diet with the 0.5% diet, so methane production dropped a lot, 34%, methane yield dropped 29%, and methane intensity dropped 26%, right? So why that? Speculations is because of bromoform. Bromoform is very high in the seaweed, in this red seaweed species. So it may be related to why you see this drop. It basically inhibits the growth of methanogens, or those microbes that produce methane in the rumen. So in the beef cattle, also even more pronounced. So this study here was done in UC Davis, and they measured this in beef cattle. Well, they saw with the same levels of the seaweed, the same species, they observed a 60% reduction in methane production, a 68% reduction in methane yield, and a 73% reduction in methane intensity. In this case here is grams of methane per kilogram of gain, average daily weight gain. So again, bromoform is likely involved. The closest thing that we have in terms of data with this Asparagus taxiformis in terms of grazing is a study that I collaborated with Kathy Soler again. And at this point, we did basically an in vitro study using that fermenters, as you can see here, that simulate the rumen environment. And we fed up to 1.5% of Asparagus in the diet. And we did this because we did not, those studies that I just mentioned was not published yet. And we were following some data that was from other in vitro studies. But what happened, and you can see here on the top line, is the diet where the cows were fed, not cows, the in vitro system was receiving a orchard grass diet, right? So that's a pasture diet. And we supplement the pasture diet with the seaweed. There is no grain in this diet, and basically simulating a grass-fed system. What happened is methane dropped almost to zero in 3 hours after we start the inoculation process, after we add the seaweed to the system. And then as you can see, it remains at zero for long periods of time of the day, right? So it's really effective in drop methane. Again, bromoform may be involved. We don't necessarily want to obliterate methane like you see in this study. For sure we fed too much. You may not need 1.5% in the diet, but you can see that basically move methane down very, very quickly, right? So, and again, these were simulating a pasture diet, right? Another mitigation strategy that's out there is the 3-nitroxypropanol, or 3-NOP. This study was done at Penn State. And 3-NOP, again, 3-nitroxypropanol, is a product that's not yet available in the US, not been approved by FDA. However, in other countries, like I know, like in Brazil is approved, and I think in Chile has been approved, and also seems very promising. You can see here in this study that was done over 12 weeks, which is kind of good because it's relatively long. The control diet here is in the top, and the cows were supplemented with different levels of NOP, a low, a medium, and high dose. The dose are in the bottom here, you can see. So you can see that drop methane production in approximately 30%, right? When you look at methane intensity, grams of methane per kilogram of age-corrected milk, kind of the same trend, about 20% reduction, between 20 and 30% reduction in methane intensity. So again, it's promising, but it's not yet approved in the US. And then to kind of finalize here, Larry mentioned those comparisons between 1940 cow and a 2007 cow, and then a 2007 cow with a 2017 cow. One thing that we know, the US dairy industry, it has made impressive change, right? If you see this data here from USDA NAS, from 1970 to 2018, you can even, easily find the data up to now, actually, up to this year. But so you can see here in the orange line, that's the milk production. And in the blue line, that's the number of cows. So the number of cows dropped almost from 1970, you have about 12 million cows. Right now, we have around 9 million cows, right? So about 3 million less cows, and milk production increased almost double in the same time frame, right? From 120 billion pounds of milk produced in a year here to about 220. So this basically shows the improvements in genetics, animal health, technologies on those dairies, and nutrition as well, right? So to summarize, the meta-analysis that we did show that cows that are grazing, they eat less and they produce less milk and milk components. However, but they have similar estimated methane yield and methane intensity, and also similar milk nitrogen efficiency. However, I want to mention here that this data should be interpreted cautiously because we have a low number of treatment mean comparisons, only 14, right? There's some promise here with forage canola, the seaweed, Asparagus taxiformis for sure, the red seaweed, and also the 3-NOP, right? They show substantial reductions in methane production, yield, and intensity. So those are promising feeding strategies in the future. I mean, it's ongoing, but in terms of future research, I think we need long-term feeding trials, specifically with this red seaweed. Those studies that I show, I mean, the study that I showed with the dairy was a short-term Latin square design type of study. We need continuous type of design with feeding seaweeds, with the red seaweed in grazing, not only in confinement, but also in grazing cows, right? There are limited information regarding the use of seaweeds and 3-NOP on grazing dairy herds. I think that as far as I know, there is no studies with the NOP in grazing herds. I may be wrong, but I think that's the case. So we need more data on this for sure. Also, scarce information on how seaweeds and 3-NOP can impact animal health and also consumers' acceptance. And also, I think that further research is needed to either to optimize current approach or identify new approach that you can simultaneously decrease both methane production and intensity in a cost-effective manner to produce. That's key here as well. So with that, I'd like to acknowledge some of the funding source of some of the studies that I show here. Also, Jennifer, for the invitation to participate. And now we can open up for some questions. Thank you.
Andre, that was fantastic. I give a resounding applause to both you and Larry. Thank you so much for sharing your work today. If we're going to post a poll for everybody to just respond, if you could do that, that would be great. And I will start with the questions. The first question is from Brett Chedzoy. Is the red seaweed the same as the ground kelp supplements readily available as a livestock supplement? If not, do you know if kelp has a similar benefit?
No, it's not the same. So the kelp that is available, basically a lot of companies sell kelp, they call kelp meal. It's from another seaweed. Actually, it's a brown seaweed called Scophila nodosum. We have done studies here at UNH with kelp, and data are not promising. In one grazing trial that we did, it only reduced methane. We measured throughout the grazing season, and only changed methane early in the season, and the reduction was quite low, about 10%. We follow up with another study that was done during the winter. The cows were in confinement, and we fed quite a lot of that kelp meal and did not change methane emissions at all. What I can share in terms of the seaweed, we're testing another seaweeds called Chondrus crispus, which is available locally here in the Gulf of Maine. And that one seems a little bit more promising. It's also red seaweed and drop methane up to 15%. So, but we need more studies on that one.
Okay, thank you. So Dickon Crane asks, does the carbon emissions from grass-fed cows account for the emissions produced by the grain production compared with the grazing? Are other offsets like transportation emissions accounted for?
Well, I would think that is accounted for on those scenarios, because all those scenarios that I show do have grain being fed to those cows. So as far as I know, with the IFSM model, the integrated farm systems model, it does account for grain production. Yes, and transportation as well. I mean, transportation, I'm not sure. It accounts for the farm equipment and farm tractors and equipment. I know that account.
And I will just say on sort of the national and state inventories, those are accounted, but they're accounted in different areas depending on how the inventory accounting scheme is organized. So the next question is from Kirsten Workman. Would 3-NOP be allowable under organic certification programs?
I don't think so. And I mean, because it's a synthetic product, I think it's basically, as far as I know, the way that inhibit methane, it's via competitive inhibition, because it's like a enzyme, right? It inhibits the last step of methanogenesis. It seems is a synthetic product, and as a synthetic product is not allowed in an organic system, which is which I guess in one way, unfortunately, but because by definition, organic farms need to graze, right? But to be certified, and the cows must consume at least 30% of the intake coming from pasture. So I think overall, grazing has fewer options than confinement, but there's, I'll say, not only in the US, but maybe worldwide, you know, there's way more grazing cows than confinement, if you really think about it.
Fiona has asked, how easily will it be to convince dairy farmers to produce or to purchase and adopt novel feed supplements? Do you foresee voluntary carbon markets helping with adoption?
Oh, I would think, you know, what I can say, some of these products that are promising, like the red seaweed and the 3-NOP, would add costs, right? Right now, the red seaweed, it seems, well, one thing, it's wild harvest. It's really expensive. It's produced in Australia or in Portugal. I know there is some groups in Hawaii, that some companies that are trying to grow that in an aquaculture system. There is issues in terms of biomass. It's highly invasive. There's a lot of things going on there on the Asparagopsis end of things, and it's expensive. The 3-NOP, I don't know how much cost is, but I think the bottom line is, if the farmers, if there is any incentive, like you just said, carbon credits and things like that, I see they adopting, but at the moment where they basically, you know, dairy farmers are struggling just to, specifically medium and small size, to keep alive or keep producing milk. I think it would be more difficult if there is some sort of incentives like carbon credit or something along those lines to subsidize some of this.
Okay, it is 11 o'clock. I have one burning question myself for you, but we have officially ended this meeting. Thank you all for joining us. You will see that the recordings will be posted 7 days from today on our website, and the link is in the chat box. And we have more webinars coming up: February 8th, manure management and greenhouse gases; March 8th, field nitrogen and greenhouse gases; April 12th, to be determined; and May 10th, food waste and greenhouse gases. And you can register for those in that link to the side. Thank you, Larry and Andre, for joining us. And if I could just ask my one burning question, Andre, before you go. So the archaea are only 1% of the rumen in terms of the population. They're methanogens, and they're the ones who are making the methane. But they're providing a service to the gut of the rumen, which is they're a sink for the hydrogen that is produced from all of these other processes. And so that is presumably keeping the rumen from becoming extremely acidic, correct? So we can't knock out those archaea because they're providing a really important service to the health of the gut. And Larry made a point in the beginning that was really great about how, you know, if you have methane, you're not properly using up all of your foodstuffs, right? So I'm just curious, what are some of the things that we should be thinking about when we are considering adding feed amendments to the diet, to think about both the well-being of the cow, as well as what ends up in the manure, which then ends up on the field? Like, how should we as a society be thinking critically about how those feed amendments are affecting the ecology in the rumen in order to reduce that methane, so that we aren't being foolish, and also understanding that bacteria, you know, they double every 20 minutes or every 2 days, depending on the species, and they're highly evolving creatures, right? So how can you share with us your thoughts on how we can be sensitive and thoughtful?
Yeah, I mean, well, there are a couple of things here. Well, one thing, as you said, we cannot eliminate methane just because it's a natural product, and just by evolution, right? Millions of years, that's why they're there, feeding that ecological niche there for the cows, basically disposal of hydrogen so the fermentation can keep going. So in fact, I feel that I guess that the point here is not completely eliminate methane. It decrease methane to levels that we can, let's say, 30 to 35%. If we achieve that on a cow, 30 to 35% reduction in methane consistently means, even though some of the studies that we show here that measure to 12 weeks, that's not the full lactation cycle. Of course, doing full lactation cycle studies is very expensive, but that may be some of the needs out there. So to really test this in the full lactation cycle. So one thing is persistency, right? It's very important that consistent keep things down, because microbes may basically, as you mentioned, evolve, and then at the same time, they may not be more sensitive to some of those or bromoform or glucosinolates or 3-NOP. So we don't know that. So we need to do more long-term studies. So the other aspect is the cost effectiveness and application of those. So those need to be considered in any way, shape, or form that we want to design strategies to mitigate methane. You know, I guess that just to summarize, I think we should look for reduction mitigation that are consistent and important, like levels of 30 to 40%, if that's, you know, and consistently, and cost effective. You know, I feel these are important targets. I know they're difficult to reach, but should be considered in also doing some of these long-term studies.
This has been a production of Cornell University, on the web at cornell.edu.